Hidden markup models are used for different applications in biological domain, but still they have their several shortcomings. For example, one common shortcoming is that they do not deal well when there are correlations between the nucleotide sequences. So what are these correlations? Basically, most common correlations are long range correlations. So when those nucleotide sequences are aparted and their distance is high enough. Similarly, one example can be for RNA base pairs, which are also conserved regions, and when those conserved regions of RNA base pairs are long ranged and their distance is usually high. So in those particular cases, HMMs will not produce very good results. Similarly, if training data is low, HMMs will not produce good results. So training data means those data based on which we are training our model. So if that training data, number of those instances or number of those words or sequences is small, while training that HMM, functionality of HMM would not be very good. Because with the increase of size of the data, HMM parameters converge to a very optimal position. So what are the advantages of, or what does a good HMM stands for, or what makes a good HMM? There are two things. Number one, it is very good for classification problems such as scoring of proteins which are used for protein family modeling. And the other thing is annotation of annotated or unknown data such as gene finding. So they are very good in those circumstances. So what are other parameters while we are training any HMM? Number one, our observation sequences should be very clear and have some meaningful information, and their tags should be very good. They should not be confusing tags. Then unordered observations are not good for training HMM. It means that whenever we are putting some observations for training of HMM, they should be in some specific order instead of unordered observations. Again, it is beneficial, but it is not necessary that observations follow some grammar or some set of rules. So, but for guideline, we always say that there should be some specific guidelines that which observations should be put in training for a hidden Markov model. So if these observations or guidelines are present, then it is very easy and straightforward and fast for training a HMM for our provided data. So based on this information, they are then used for gene finding or protein model generations. So here I will give a list of 3 most common HMM tools for our biological data. Number 1 tool is Whale, which is basically based on grammatical structure and a very modular design. Modular design means that there are separate states for codons and non-coding regions, which were our introns, exons and supply sites. And then other advantage of this module is, or this tool is basically, it uses many states. So with the increased number of states, working of HMM also works. Why? Because each state is performing or emitting only one symbol, so there are very less chances of confusion or complexity of the model. So Whale basically it is a very simple model with many states available, and then these states, what does they do? They provide independence between individual states. So these states are shown by different symbols in this diagram, which are A, C, G, T, and again A, C, G, T on the second level and so on. So we start from the start codon, then we move toward the first level, which is on this thing, which are having 4 states, A, C, T, G, which are our 4 basic symbols. Then in the second level, we have again these states, which are 6 in total, and then we have 6 states here. So in total, 16 states are here with different combinations. So we have start codon, then 4 states, and then remaining states, and then finally we have this final state here. So this Whale model, it is very simple and having large number of states, and it is used for gene finding and other applications. Similarly, there are 2 other models or software tools which are common. Number 1 is HMM Gene. It basically extension of hidden Markov models with just one more module, which is for classification, and this is called CHMM. C stands for classes, of course. So what it does do is it is basically it takes helps from other statistical algorithms in relation to hidden Markov models. So it uses high order states, और इसका एक और advantage ये है कि it is very very fast. Whatever data we have, it takes all the data in one go and then trains the model against that data. And then finally we have another software package which is called Ghani. So Ghani is basically a generalized HMM. It can be used for several purposes. One purpose is of course for biological data modeling. And what is the structure of this? We have different edges, and then these edges are basically itself complete HMMs. So we have one edge, so it is HMM, H2, it is another HMM, and then these edges are connected together. And then in each edges there are different states which can be of any arbitrary or random types or random order. And then these states themselves are again based on some other statistical frameworks such as neural network. So those neural networks work together to generate some high output result. So basically this is a generalized model which can do several tasks.